Okay, here we are. So the last segment for our group is my talk, which is on the Borrelia prevalence in wildlife hosts in Atlantic Canada, and specifically in New Brunswick, where Mount Austin is located. So this was part of my master's that I did under Dr. Vett Lloyd, and this research was mainly conducted in 2016, 2017. So we've heard a lot already about Borrelia burgdorferi. That is a very common species, and it's the one that gets most of the attention for Lyme disease. The other Borrelia that I was interested in was Borrelia miyamotoi. So this, well, there's some argument about this today, is in the relapsing fever group. So symptomatically it presents differently than burgdorferi. But what's most important about it is that it was only very recently recognized to be zoonotic. So the first human case was recognized in 2011. So it has been around for a while, but it's much more recently been identified as another zoonosis. We now know that previously it's actually been getting misidentified as burgdorferi. So with these two Borrelia species, they share this all-important vector-host transmission cycle. So for most people, a lot of the attention gets focused on the vector, which in northeastern Canada is Ixodes scapularis, the black-legged tick for both. But of course, equally important, if not in some instances more, is the host, as not all hosts are actually equal. Some species are what we refer to as reservoirs, which are animals that are actually much better at maintaining and competently transmitting that Borrelia infection to new generations of naive ticks. So some examples that are found in New Brunswick are the white-footed mouse and the eastern gray squirrel. So again, these animals are really good at transmitting, and they're much better than other animals at actually maintaining this infection. Where burgdorferi and miyamotoi do differ is that miyamotoi has the ability to vertically transmit within ticks. It has been documented that in infected adult female ticks, the spirochete can pass to the eggs, whereas this has not been shown at all with burgdorferi. So while they share generally the same transmission, miyamotoi also has been shown to have vertical transmission in ticks. Now, my project started with a very simple idea. In New Brunswick, we had a lot of information on human incidences of Lyme disease, on tick infection prevalence for Borrelia burgdorferi, and we knew nothing whatsoever about the wildlife. So there was always this issue where people would wonder whether or not these infected ticks were adventitious, being brought in from the US, or if we actually had the full life cycle occurring in our province. Thankfully, it's very easy to test that. You just need to actually find a grad student to go and look at the animals. So we knew we had reservoir competent species. The next step was to actually look at whether or not they had Borrelia, in which case they may actually be contributing to the tick levels we are finding. Since this was literally the first look, I set no restrictions on what animals I was going to grab, and anything I could capture, I would test. This was everything from migratory birds to rodents, and if you've never been to this area of Canada before, lots and lots of porcupines. So there was two methods to this collection. The main thing I was doing at first was collecting roadkill. So this was an easy way to get medium-sized animals and have, very nice GPS coordinates for where the animal was found and lots of good information on that. But of course, in order to increase the sample size and to also target small mammals, we started the cat kill program, which was a fully public participation, community-led initiative to get small animals, mice, birds, and the like, brought in and sent to me for testing, which also was wildly successful. So through this roadkill and cat kill method, I got all of my samples to test. And for every single animal, what I was testing was 2 tissues. So I targeted the liver and the kidney to do all of the downstream testing I'm going to be talking about now. So as you've heard with Samantha, I was also using nested PCR. So at the time in 2016, the surveillance standard that we had in Canada for ticks was to use 2 genes for Borrelia burgdorferi. There was no standard for miyamotoi because it wasn't something that was being frequently sought after or investigated, especially in Canada. The issue I came across is that when you try and take these primers and throw them into every animal that you find, most of which that don't actually have their genomes available, you end up having some issues. Thankfully, using the 1623S IGS primers, so those same primers that Samantha mentioned, I was able to set up a working protocol to use in my animals. So these primers came out of the National Microlab of Canada, but what I did was just make a new set of inner primers for the nested protocol that were species specific. So one that would detect Borrelia burgdorferi and one that would detect miyamotoi. And of course, these went through rounds of testing for specificity and sensitivity. Once I got all of this testing done on all these animals, the first thing to note is that if I found one tissue positive, that counted as an animal being positive. I did not differentiate between there being a single organ or two positive. But all told, I got 339 animals, the overwhelming majority of these coming through the public donation program. These were split across 29 species and not split very nicely. So if you look to the table here, my most prevalent animal that showed up was the meadowvole, which also just makes sense in terms of the species composition in the area, but this is a small rodent. And then, of course, lots of deer mice and shrews, and then also, similarly, a lot of porcupines. The most interesting find for this was the jumping mouse, Napiozapus insignis, which is a small rodent that's not been investigated for its reservoir potential that much, but it had high levels of Borrelia burgdorferi and miyamotoi found, just 2 and 3 respectively, but out of only 21 mice sample. Next, of course, we had the meadowvole, the deer mouse, and the eastern gray squirrel. These are all animals that are known to be competent reservoirs for burgdorferi that here in New Brunswick are shown to also carry the infection. The eastern gray squirrel in particular, it was found to have miyamotoi, and squirrels have been cropping up a lot in miyamotoi research for the role that they play in reservoiring it. In general, burgdorferi was found more than miyamotoi, which is exactly what we would expect, but the next step was to investigate further. So there are a lot of ways that people will look at the risk and the prevalences and how they can maybe map that and turn that into useful risk information for people. One of the easiest things to do, if you can, is to try and use a species as a predictor, because it's a lot easier to track a small mammal than it is, say, a population of ticks. So if one species is more likely to carry the infection, that can be used as a predictive factor to help with public policy. Unfortunately, because my samples were so broad and the sample sizes were so disparate between species, I wasn't able to get any significance here. But just finding and identifying these positive species within the province was in and of itself a very significant find. The next thing is, of course, especially in terms of public health people, a lot of these decisions for risk are made based on geographic regions. So of course, it'd be interesting to look at whether or not we were showing some sort of spatial correlation for these positives. So this was a simple hotspot test to see whether or not our positives were clustering. And for both species, burgdorferi and miyamotoi, that wasn't found to be the case. As I'm about to show you a map of my samples, what ended up happening is we had some incredibly dedicated people donating to our samples, and we ended up having a very clustered distribution of our actual samples coming in the first place. So this map here, if you're not familiar with New Brunswick or Canada, shows in the small inset the 3 main cities of New Brunswick. So we have Fredericton, St. John, and Moncton, with Sackville and Mount Allison being a little bit to the right of Moncton. So New Brunswick borders Maine, Quebec, and Nova Scotia. So it's surrounded by provinces with lots of endemic areas and high incidences of Lyme disease. All of my squirrels came from within the city limits of Fredericton, including the Borrelia miyamotoi positive. So the gray squirrels just happen to have a more western distribution within the province. And again, the positive squirrel was found within the actual city itself. The vast majority of my other samples were in the Tantramar region, which is the area of Moncton and Sackville within New Brunswick where we were located. And so you'll see that, of course, we have a lot of our positives here, both the roadkill and the catkill, but this is highly clustered with a lot of our samples coming from Sackville itself. The next step, though, is after looking at where the bacteria was in the province, is where it is in the animal. As many people have looked into, the actual spread and distribution of Borrelia within a host after an infection is a very difficult topic, as it seems to be almost random. So I took my positives for burgdorferi and miyamotoi and decided to test every organ that I had available to me to look at where the bacteria was present. So I took all of these animals and then as well as some species and location matched negatives and tested everything that was available from them. Looking at just the singly infected animals now, not the one co-infection that occurred, what you're seeing is just a truncated table of what ended up being positive. So you see it's a very small amount of tissues shown, but the first thing I'm going to draw your attention to is that the liver is the most prevalent, and it was one of the initial 2 testing tissues. And the second thing is that there's only one diffuse infection where a Borrelia was detected in multiple organs, and that is within the meadow that had Borrelia miyamotoi. The next thing I will point out is that 2 of these animals in this table are listed as negatives. I actually found more positives when I did this testing, where the Borrelia was only detectable in the brain and nowhere else within the mouse. So that find was pretty interesting in itself, finding it in the brain, but what was more interesting, through sheer luck, was the co-infection. So this one co-infection was a pregnant female jumping mouse. So it had tested positive for both Borrelia, and it also had 2 fetuses recovered that were in late stage of development. So in addition to all of the maternal tissues, the 2 fetuses were recovered by microdissection, and their head, gut, and placentae were treated as different samples and tested. All of the fetal tissues and most of the maternal tissues were miyamotoi positive. So again, in the maternal brain, heart, etc., and in the fetuses, Borrelia miyamotoi is detected. Borrelia burgdorferi was only detected in the liver, so it was still confined to a single organ for detection. In general, as Dr. Lloyd mentioned early on, this was nested PCR detection. This was direct detection of the DNA. So this shows that the bacteria is present in the tissue when it is being tested, as opposed to indirect methods like serology. I, of course, found that liver was best for detecting both Borrelia, but this was an initial screening tissue based on some a priori knowledge. What was interesting was how many positives I ended up finding in the brain. The brain was never something that was really looked at as being a sampling tissue, and it leads to more interesting theories about the possibility of a refuge. So in the first objective, it was quite simply, we know we have Borrelia in our ticks. We know we have Borrelia in the province. Do we have it in the wildlife? And then, of course, do we have this full vector host reproduction cycle of the Borrelia happening within New Brunswick? So I made the methods, got the samples, and found that we do in fact have Borrelia in our non-migratory and reservoir competent species. We still don't know to what extent the specific species are contributing to these levels, but that can come next. The wildlife levels do reflect the tick infection data, so there was nothing unique to be found there. In the second objective, looking at the extent of infection, the liver was the most commonly infected tissue, with the brain being the second most common. The fact that almost all of the infections were single organ infections, again, suggests that there might be some refuge mechanism happening where the bacteria is sequestering in a tissue. But most interestingly was the finding of the jumping mouse fetuses. So in these fetuses, Borrelia miyamotoi was confirmed present. We know that within ticks, vertical transmission is possible. Maybe this suggests that there could be the possibility of that within some mammalian species, but it's important to note that there's no vertical transmission officially unless there's a live, viable birth. So now I believe Dr. Lloyd is going to come back to continue the discussion.
I will, right after I unmute. So the last, to avoid us fiddling around and changing slides, I'll just talk, and Chris, if you don't mind advancing the slides at semi-logical places, that would be great. So the global conclusions are that we're not just looking at one reservoir species, there are multiple reservoir species. And I do realize that most people around the world, particularly those in urban areas, are not going to be overly worried about whether or not the black bear that has just bitten you has Lyme disease or not. Besides, if black bear bites you, Lyme disease is not your most immediate concern. But there are many species which harbor Borrelia and seem to have the bacteria live in them quite happily without becoming sick enough to actively die. So there's a diverse source of potential bacteria, sources of bacteria in the environment. Perhaps an even more important thought is that there are a lot more Borrelia out there than Borrelia burgdorferi. We know that, but we're only just now starting to see what's present in the environment. And this is important for a couple reasons. The first one is, as Chris just presented, different Borrelia species show different tissue tropisms in the body. Borrelia miyamotoi is much more widely dispersed in the body than Borrelia burgdorferi in wild mice. It's important to emphasize that. Obviously, the question is, for those interested in human medicine, is this also true in humans? In terms of the significance, I want to touch on Samantha's work with leptospires. Leptospires are a somewhat basal group of spirochetes, and these spirochetes are shed in the urine. The adaptation to the tick as a vector is supposedly a Borrelia-specific innovation, but that doesn't necessarily mean that Borrelia spirochetes have lost their ancestral ability to invade epithelia, particularly bladder epithelia. They're present there. It's in fact a very good place to find them, and there is evidence that they're shed in the urine, extensive evidence actually. So we need to have an open mind when we're talking about transmission. There's maybe more happening than just ticks. The other rather worrying thought is if Borrelia burgdorferi is one of the most tick-specialized species, and there is some evidence for that, it may not actually be a good model for everything that's out there in the world. In Europe, at least thankfully, you study more than Borrelia burgdorferi, but really people are only just beginning to appreciate the diversity of Borrelia in the wildlife in Asia, in Africa. So if we're just, it may be that of the bulk of the studies which have been done on Borrelia burgdorferi, and in fact one strain of one species, have been done on an atypical species. And this is incredibly important because the impact of borrelioses on individuals, families, and communities is so significant that we really need to get this research right. One good tool for getting it right is to partner with communities and with patients. It means better research, and our research, that way we can be sure our research meets the needs and the goals of people in the world. So with that, we actually have a few minutes for questions. So thanks to everyone who's helped, citizen scientists and their cats. And if Chris, if you go to the next slide, happy to take questions, comments, what have you.